Fantastic. If everyone could please take their seats, we have an action-packed session today and this morning. Fantastic. So doors are closed, coffee's in the system. So it's a great pleasure to introduce this session. My name is Maya Ladd. I'm a neurology trainee from Newcastle, and we're joined by Professor Rita Horvath from Cambridge. I was thinking about how to introduce this session. I got reminded about all the colleagues and medical school students that were talking to me and asking me why I wanted to do neurology, because I couldn't do anything for my patients. Well, little did they know about what the future would hold and the sort of promise it holds, and particularly in terms of this group of disorders that we'll hear about today. And so I had great fun pinging all the invites, despite them being surgeons and oncologists and whatnot. I didn't get any replies. Our first talk is by Professor Gabriele Lignani from the UCL Queen Square Institute of Neurology. He's a professor of translational neuroscience and works on gene therapy and editing techniques for neurological disorders. So Gabriele, take it away. Thank you so much.
Thank you very much for the introduction, and thanks for the invitation. Today I will speak about gene therapy for epilepsy. And if someone tells you about something about gene therapy for epilepsy, you think that it's something related to I have a mutations in a gene, I want to correct it. In this case, it's a bit different, and when we speak about brain, also it's different. Because, for example, if you think about ion channel mutations, this mutation that happens very early in life will modify the circuit in a way that at the end the brain will be different. So it will be very difficult to just correct the mutations after when we know that when we have the possibility to correct it, so between neonatal and adult stage. So there are many ways on doing gene therapy, and in this case the question is, should we treat at this stage, should we treat the cause, so correcting the mutation, for example, or we should treat the symptoms? So there's been done a lot of work so far, also in my lab, both to develop therapies for treating the cause of the disease, and some are also in clinical trials. But there's been done a lot of effort as well in trying to treat the symptoms. And I will speak about this today. So, because it's a very diverse audience, I thought to give a bit of an overview of why it's important to treat epilepsy. So, it's a bit unmet need. 1% of population has epilepsy, so more or less 50 million worldwide. 2.4 million every year diagnosed with epilepsy, and is the second most serious neurological disease after dementia. And most of one-third of the patients are drug resistant, so they have no drugs available for them. These kind of patients often have focal epilepsy, so the 70% of these one-third, so millions of people, meaning that the epilepsy starts in a precise region of the brain. For these patients, the best option is the brain surgery, so remove that part of the brain. So we thought how we can leave out this option to take out the part of the brain, and also you have to think that it's only available for a small portion of this, so there is a lot of millions of other patients that have no options at all, not even the surgery, because, for example, if this brain area is very close to eloquent cortex, you cannot remove it. So the idea is that if you have an epileptic focus defined in the brain where the seizures start, instead of removing that part of the brain, we can use gene therapy, so injecting a viral vector there that stays in that region, and we can modify neuronal properties. And as you know, epilepsy is too much excitation in the brain, so the idea is we can modify neurons, decreasing their excitability. And we know how to do that for decades of study of biophysics, knowing how to modify and modulate ion channel, for example. So up to now, there's been many groups trying to develop the gene therapy in this way, trying to decrease excitability in the epileptic focus, starting from optogenetics and chemogenetic approaches. There are many, and today I will focus mostly on modulating endogenous channels. So try to modify the expression of channels that are expressed in neurons to modulate their excitability and decrease the overall excitability in the epileptic focus. So I will speak mostly about A channels, KCNA1, that codified for the potassium channel Kv1.1, that I call as well EKC, that means engineered potassium channels, because we modify these channels, modifying a single amino acid to accelerate the recovery from activation, so to make it even better than the endogenous one. And we show in many studies that overexpression of these channels can reduce neuronal excitability and can suppress seizures in some models of epilepsy. So what we wanted to do next is, these models are very good, but we wanted also a more translational model, and we want also to see if we know the genetic cause. There is a genetic cause, because these models that we published in the last years were induced models of acquired epilepsy. So we want to see if we have genetic mutations that cause epilepsy, can we still use this approach of decreasing, suppressing the symptoms, so treating the symptoms of suppressing excitability can work also for these kind of models. So what we have done, we established in the lab a model of focal cortical dysplasia that is due to somatic mutations, so very few neurons have these mutations, and cause a small region of the cortex where that is not well developed, so it's malformed, and create like this lamination and also different neuronal subtype like giant neurons. And it's known that the seizures start in that smaller region of the brain. So we decided to model that in an animal model. So we can have genetic models that cause focal epilepsy. And it's very important because it's the most common structural brain lesion in children with the drug-resistant epilepsy. It's the most common cause of a surgery in children, so it's quite a big unmet need. And more than 50% of these of patients with focal cortical dysplasia, the cause is mutation in mTOR pathway. So what we have done, we established a mouse model with a neutral electroporation. We electroporate in a small proportion of progenitors an activated Reb that is an activator of the mTOR complex, so creating this hyperactive mTOR complex that is the basis of this pathology. We show that these neurons, they should be in the layer 2 and are all in different places. We show that you can have, like similar to the pathology, giant neurons, and as well you have an increase of the mTOR activity. So this is very focal, and this is in the prefrontal cortex. So we had this nice model that is genetically linked and that has focal epilepsy. And then we want to test, first of all, we characterize the mouse model. We show that 65% of these animals electroporated with these constructs develop spontaneous generalized seizures, and they have also behavioral defects like memory, and also olfactory habituation is impaired, mostly when there are social cues, because obviously it's related to the prefrontal cortex. And also we show that the animals with the seizures, so the 65%, has more severe phenotype than the one that don't develop seizures. So once we established this model, we created a preclinical study in which we developed the model, we implanted transmitters, wireless transmitters in the mice, and did some behavioral tests as well. Then we record the baseline epilepsy activity, and then we inject in that particular area where the seizures start our viral vector with the potassium channels. In this case, we use the CAM kinase promoters to drive the expression, to excite the neurons there and decrease the excitability. So then we wait for the virus to incubate, and then we record again for 2 weeks to see the effect of our therapy. So what we saw basically is that this is control animals, and this is the animals with injected with the CAM kinase, EKC, the potassium channels. This is the first part is before the therapy. The orange line is 2 weeks waiting for the virus to express, and then after the therapy. Basically, if you want to analyze the data, we saw a clear decrease that compared to the controls is around 80% decrease in number of seizures when we, in the animals treated with our therapeutic approach. And we saw an increase in seizures-free day. So basically, this means that we can rescue and we can decrease of 80% the number of seizures in a model that is due to genetic mutation, but anyway cause epilepsy. So we can use this approach for many different pathologies that leads to epilepsy, no matter what is the genetic cause. So as I say, there are many approaches. All these approaches, including the one that I show you, as part from is based on a concept that is that if you have a physiological brain and then this is the epileptic brain with more excitation, you can just decrease the excitability of the excitatory neurons, as I say, using, for example, CAM-kinase promoter. So it's a bit a simplified way to see the problem. So you have more excitation, I decrease the excitation in all the excitatory neurons in that epileptic focus. But actually it's not working like that, and the brain is a dynamic system as well, the pathologies. And there's been many studies, both in mice and I show in humans, showing that not all the neurons there are hyperexcitable. So not all the neurons has to be treated, not all the excitatory neurons are part of the pathology. It has been shown in mice that with the repeated seizures, some pathways, some neurons are activated, some not. And has been shown also in humans that actually seizures after the other seizures in humans with probes in the brain showing that the single unit, showing that some neurons in the seizures are activated, some are actually, there are decrease of excitability in some neurons. And it seems very reliable between one seizure and another. So there are some neurons that are more excitable, some neurons less excitable. So it's a bit a simplification thinking, just we decrease the excitability of all the neurons there. And also the changes from a physiological brain to an epileptic brain, that means also that there are different states in that epileptic brain, because there is also non-seizing. Most of the time patients are not seizing, and probably 1% of their time they have seizures. And there is a multidimensional changes. It's not just that you have more excitation. There are anatomical changes, synaptic acceleratory features that change. The transcriptome is changing. So it's not so simple as I have more excitation. Plus, one of my favorite topics, there are also homeostatic compensation mechanisms that probably are still there. So when you have a therapy that is constitutively expressed to decrease excitation, maybe there are mechanisms that try to compensate for those. So all these two slides is just to say that it's not so simple as you think. The gene therapy has to follow how the brain works and how the pathology works, and not has to be constitutive. And I think that not the problem, but is more historical issue, is that the gene therapy in general comes from other organs, like liver, in which cells have more or less always the same they have the same features and they have the same sort of activity all the time. The brain is more complex, as we know. So there are dynamic changes all the time. So in my view, an ideal gene therapy is the one that focus only on some neurons. So if you have a physiological state with obviously some neurons are more active than the other anyway in a normal brain, in a healthy brain. In epileptic state, you have more neurons active. The constitutive gene therapy will decrease the excitability of all the neurons, also the ones that were not part of the pathology. So the ideal gene therapy is the one that just correct and decrease the excitability in those neurons that are hyperexcitable. So, but how we can do that? So which neurons we need to target? We don't know which neurons are more active, probably, and between each individual will be different. And so how we can distinguish between healthy and unhealthy neurons? And how a therapy can follow neuronal dynamics. So the idea was, can we try to target only those hyperexcitable neurons when it's needed, when they are hyperexcitable, without knowing which are them? Just to try to find a way to just say, yeah, if you are hyperexcitable, I decrease the excitability only of those neurons. So we thought that we can take advantages again on previous work done, as I said before, with biophysics, in this case with molecular biology, and we know that there are genes, called immediate early genes, that respond very quickly to change in activity. And everyone knows, for example, the CFOS gene that has been used for many studies to show that a cell is active. So what we decided to do, that we published at the end of last year, and it was the work of my PhD student Yichen, is to try to create a closed loop, a genetic closed loop in the neurons, in which we used a promoter like the CFOS promoter, an immediate gene promoter, that drives the therapeutic transgene, the potassium channel that I showed before. So basically, this system is off if the neurons are not hyperexcitable. When some neurons become hyperexcitable, the immediately genes increase the expression, and this promoter that we put in our construct will drive the therapeutic transgenes that in turn decrease the excitability. So basically, it's a closed loop inside the neurons that decide if a cell is too excitable, the tool will decrease their excitability. So basically, I put it here, we create this closed loop that is both a closed loop in time and space, meaning that we don't need to know which neurons is more active and when. We just put the tool there, and the tool decides by itself when the neurons is hyperactive and decrease their excitability, and when do that. And it's not just seizures, it's not just the seizures that drive the expression. We show also that intellectual activity, so the pathological epileptic activity that happens between the seizing time, is still activated this tool. So you can basically, what we think is you can reestablish a normal circuit dynamics in the brain and stop the seizures. So we developed a tool, a protocol to try to understand the features of this closed loop genetic therapy. And basically, we use wild-type animals. We inject our virus in wild-type animals in the hippocampus. Then we inject a single dose of proconvulsant that we set it in a way that it can elicit only one single seizure. And then we check the severity of the seizures. Obviously, in the first time that we inject the proconvulsant, we don't see any difference between the controls and our therapeutic construct because it was not activated. It's our wild-type animals. And then after 24 hours, we inject again the same proconvulsant to have a second seizures. And in that case, actually, the virus should be on because it was activated by the first seizures. And indeed, we see a decrease in severity comparing the controls with the therapeutic construct. And then we wait other 2 weeks that we thought that it can be switched off, and we didn't see any difference again with the third seizures. So basically, this is the quantification. And basically, what we show with this experiment is that this tool is not active in normal brains because we don't see any difference at the beginning. It's activated by seizures because we saw the effect in the second seizures. It treats activated neurons because the neurons that were activated in the first seizures is helping to decrease the severity in the second one, and plus it's transient because if we wait a while, is not going to be activated anymore. So it's a genetic closed loop, transient genetic closed loop. And then we checked in a model of chronic epilepsy. We induce a chronic epilepsy, then we record the EEG for 2 weeks, then we inject the virus as before, and we record again 2 weeks, and we saw around 75–76% decrease in number of seizures in this mouse model, meaning that is working on decreasing the seizures. But we saw an overall representation of brain function. We decrease also this interictal activity between seizures, a restore of power in the brain, and we also show that if we challenge the mouse again with another epileptic seizures after having chronic epilepsy, the brain is going back to more physiological state because the mortality, the survival rate after that generalized seizures is similar to wild type with the treated animals. And with the control virus of epileptic animals, the animals start to die with this insult on top of their chronic epilepsy. So we have some hints that the brain is more normal after our therapy. Then the last things that we've done is to test if it can work also in a human background. So we create in the lab an assembloid model where you can assemble basically 2 spheroids, one that comes from cortical human IPS-derived neurons and one from subpallian spheroids neurons, because the spheroids the subpallial will be inhibitory neurons, the cortical excitatory neurons, then you can fuse them show that they migrate in the cortical side, so a proper physiological in a dish cortex, a human cortex. We saw that is connecting the network, are firing action potential, these neurons. And what we did, basically, we put these viruses, the control and our treatment virus, in this assembloid after more than 200 days in cultures. And then we stimulate there with the proconvulsive, in this case, potassium chloride. And we saw, basically, we checked the difference in epileptic form activity in the controls versus our treatment. We saw a clear decrease of epileptic form activity also in human organoids, meaning that also the human background, in the human background, this genetic tool works quite well. So in conclusion, we show that this tool is selective for hyperexcitable neurons in time and space, as I said before, follow network dynamics, and these are things that I didn't have time to show, but it doesn't affect physiological behavior. We test several behavior, including fear conditioning, because we know the CFOS is involved in that, and we didn't see any detrimental effect on behavior. It's self-regulating transient, does protect from further insult that works in human neurons. So now the two questions that we are trying to answer is, can we go into clinical trial for focal epilepsy with this tool? We have an ongoing trial that it should start actually in 2025 for the canonical classical gene therapy with the CAM kinase potassium channels, but we are trying also to propose this for a clinical trial soon. So to do that, we humanize our construct using a human CFOS promoter that we designed in the lab. We show with some experiment that I told you before with one seizures after and 24 hours, the other seizures, that you have a response to that. So also the human promoter works in mouse model with less severity in the second seizures. And we tested also the expression and the activity dependency in a human organotype slice from patients with resective surgery, showing that is activated. Actually, yesterday we showed that is activity dependent. So this human CFOS promoter we designed follow very well the human cell activity and probably is amenable for clinical trial. And yes, this was some neurons were not activated and activated after a proconvulsive human organotype. And the last point that I want to say, I spoke a lot about epilepsy, obviously it's my field, but we really think that we can broaden this approach to many other neurological diseases, because many developmental neuropsychiatric diseases are characterized by intermittent episodes of pathology like seizures, when there is pathological activity in the middle. Plus, very importantly, several of them, including psychosis, pain, migraine, Parkinson, for example, early Alzheimer, are characterized by an increase of excitability in some neuronal circuit. So what we are trying to do now is to broaden our approach to other neuropathology diseases, neurological diseases. So thanks very much for the attention. I want to thank my lab, mostly HNQ that did all the last study, and Amanda Barbanoj that did the first study in focal cortical dysplasia, all my collaborators at the UCL and also in Germany, and all the founders. Thank you very much.
Fantastic. I think we have time for one or two questions. I also forgot to say you can tweet with #BrainConf2024.
Hello. Thank you for an interesting lecture. From the concept in neuroscience of genes and behavior, that genes by their transcription products control what neurons do, that genes and behavior, from that concept, are you actually saying indirectly that all types of epilepsy should have at least some form of genetic underlying basis? Or how do you explain what clinicians would call more idiopathic types of epilepsy?
Yeah, thanks. Very, very good questions. Actually, there are two classes, obviously acquired and idiopathic epilepsies. If you are thinking, there are some pathologies that are caused by genetic mutations, and this is unknown. like can give epilepsy, like a Dravet syndrome or SN1A, but also acquired epilepsies are obviously produce also epilepsy, for example, for trauma or stroke. Obviously, it's not due to a precise gene mutations, but will cause modification in gene expression that can lead also to the epileptic phenotype for sure. In this case, what we are thinking is, instead of focusing on which is the cause, if it's gene, not gene, if it's indirectly a gene expression, we just treat the symptoms that are the seizures, because no matter what is the cause, at the end the patients have seizures. So if you can control the seizures, it's a big advantage for the patients.
Congratulations, beautiful set of experiments and the paradigm itself. Could you shortly elaborate on the specificity of the virus? So it targets the C4 promoter, or it works on the C4 promoter. How do you ensure that it only works on those neurons that are involved in the seizure itself? Because C4 is such a broad promoter with neuronal activity, to ensure that there's no unwanted side effects in the whole brain.
Yeah, thanks for the questions. Actually, we use the minimal C4 promoter that is less sensitive to changes in calcium than the normal, the endogenous C4 promoter. And we think that is not activating basal behavior because the increase of calcium is very limited compared to the epileptic activity. So we saw that it's really activated our tool when you have pathological activity, not physiological. We did a lot of behavioral tests and we showed no difference in the behavior. And yeah, that basically is that the point. And then we use the AV9, so we are transducing all the neurons in that region, more than 90% of the neurons. We are not biased on which neurons we want to treat. We transduced all, and only the one more active in the pathological way will be activated. And we show that the majority are excitatory neurons, obviously, but there are also interneurons that are more active during seizures, and we are treating also them. That is not known if it's good or bad, but in this case, maybe it's good. You don't know why an interneuron is hyperactive, and if it's hyperactive during only pathological activity, maybe it's good also to decrease their excitability. Thanks.
Thank you very much. I think we have to move forward for the next speaker. It's a great pleasure to introduce Professor Matthis Synofzik from the University of Tübingen, Germany. And he is a lead of the Translational Genomics unit in neurodegenerative diseases. And he has been leading a lot of international consortia on developing natural history studies and treatments for genetic ataxias. And today we will hear a presentation on the one mutation, one medicine, number one clinical trials approach.
Thank you very much, Rita. Thank you very much for the organizers for having me here. What I would like to introduce is a little bit a new paradigm, I think, at least in neurodegenerative diseases, maybe neurology as such. So the talk will have quite a few of conceptual points more than data points. Usually I don't like that, but it does need, also given the limited time, it does need a little bit of conceptual introduction. I would like to talk about individual patient-customized ASOs for severe neurodegenerative brain diseases. What is the idea and what is the problem? The problem is that in the rare disease fields, we have two main problems. One is we have thousands of rare, often ultra-rare neurological diseases, most of them being genetic, but given their rarity, there is very few pharma interest, very few market sphere out there, and there's still there's no interest to develop new therapies. That's problem number one. But even if there would be an interest, even if there would be a market for genetic, for example, therapies, there's a second problem. The second problem is that in our Western world, the current drug development paradigms are just not apt to ultra-rare diseases. That's because for going for marketing approval, you need to have usually a phase 1, phase 2, phase 3 trial. But this is just by the mere sheer number of inherently by the rare diseases just not possible. So how could you go for market approval for such ultra-rare diseases? So, and even if you would do so, that would take 10 to 15 years. And by this, more, in particular, the severe diseases, brain diseases, those patients are already severely affected or even dead. So how do you encounter those problems? The paradigm which we would like to suggest is to come up with a systematic, scalable approach, which might be applicable to many ultra-rare diseases alike, by targeting shared underlying biological principles. So not thinking about of diseases, but thinking about shared biological principles. And this might allow by that to target individual patients, even if just one patient per disease in the world, using the same modules of development and come up with individualized therapies, but by using the same modules, making it scalable and developing a platform approach. So hitting two flies with a stone, ultra individualized, but still streamlined. That is the idea. How could this work? Before I come to this, just one more excursus about this drug development paradigm, which we have in the Western world, and why it's not really apt to ultra-rare diseases. On the top, you see how we currently think in drug development. You have your preclinical work, you have your phase 1, 2, 3 trial, and this usually takes 10 to 15 years. What we would like to suggest is not to replace it, but to complement it, and this is what's needed for the ultra-rare diseases, with an N of 1 specific treatment and development paradigm. And this paradigm, you develop your individualized drug, in our case an individualized antisense oligonucleotide, do this much more rapidly than you see in classic drug development, and just by them, you cannot do phase 1, 2, 3 trials, so maybe we don't even need this. And go in a named patient setting and get the patient treatment and do this in a much quicker fashion. And this is then not geared toward marketing authorization, but beyond the level of marketing authorization. In the future, we might think about N of 1 platform trials, where we, for example, with individualized ASOs, aggregate up those individually treated patients in a platform trial, and then bring this to scientific rigor. That's the future, but for now, we start with named patient treatments. But this is the line of thinking we are currently complementing the classical thinking. And the thing, however, is, now, if you treat one patient here, one patient there, you will still need millions of years until you treat all those rare diseases. So the smart thing is, or the clever thing is, you need to make this N of 1, but still scalable. And this is why I think we need to focus on shared biological principles and streamline the process to make it scalable. And how do we want to do this? What is the shared biological principle? With the ASO approach we are developing is with the shared biological principle are cryptic splice mutations, be them deep intronic or exonic, or mutations in skippable exons. Those type of mutations you can target with the splice modulation ASO. And we are not only using the same biological principle, and the same principle of ASO targeting, splice modulation, but also using the same backbone, namely a backbone of an ASO which is already successful in trials and now in clinical treatment, mainly Spinraza. You know this from SMA treatment. This is now already in clinical care. And using the same principle, and even not only the same principle, but the same backbone chemistry does have many advantages. For example, it does allow, you have the already PK data on this, you know that this is provable, right? Regulatory agencies, EMA, FDA know this backbone, so they're much more lenient, of course, to accept this. And also you can serve this as a reference ASO to compare your effects to. So using this a shared biological principle, cryptic splice mutations, or mutations which can be skipped by splice modulation, using the same ASO principle, splice modulation, plus using the same ASO backbone, the same as being used in nusinersen, those are already parts of the platform to make it scalable. And if I say we, who do I mean? We have formed a European network called One Mutation, One Medicine. So one mutation, and for each mutation you have one medicine, meaning one ASO, which is an academically driven European platform led by colleagues from Tübingen, from Heidelberg, and Leiden, the Netherlands. And the overarching mission is to make mutation-specific ASO. So for really one patient or one mutation, develop a specific ASO. We focus on severely debilitating or life-threatening diseases because there the medical need is the highest, obviously. We're focusing, as I said, on cryptic splice mutations. Nano-rare, if they have enough patients to go for market approval, there's no need to do this. You can leave this to pharma. No need for us to do this as academia. And to do this, however, in a sustainable, scalable, and a time-efficient manner. As I said, we want to be quick because those are severe diseases by definition, by inclusion criteria. What is the network comprising? We have German partners, but it's all over Europe and even in the US. We have academic partners, industry partners, patient advocacy partners. So that's a network. And if you're interested in to join, Rita is part of this. So that's no closed club. We are open, we are coming. So it's really built to become larger. And we have established a pipeline. And this pipeline, to make it scalable and to use the same components, the pipeline works as follows. As an input, we are screening large genomic repositories, plus we get submissions from for example, the European reference networks, from clinicians and European reference networks. looking for suitable patients and suitable mutations. Suitable mutations, again, meaning cryptic splice mutations. Suitable patients meaning having a severe, debilitating, life-threatening disease and not yet a too advanced disease stage. We have a standardized classification and evaluation scheme for judging is a patient suitable or not. Once we deem a patient and his or her mutation to be suitable, we go along two routes in parallel rather than in series. One is a preclinical route. We develop a mutation-specific ASO for his or her individual mutation, test its efficacy preclinically, test its toxicity preclinically. So hoping to have an effective but non-toxic ASO. And in parallel, we are modeling the subject's individual natural history on individualized outcomes. So once we have an ASO, we already have an idea about a run-in natural history baseline on an N of 1 level, including N of 1 natural history progression modeling. And that's a new thing also. So it's not just the biology is new, but also clinical modeling. Doing single subject predictions is also a statistically new concept in the rare disease in the neurology field. We bring this together in order then, once we have an ASO ready, to put the patient on the ASO in order then to be able to clinically assess efficacy. And we have, as I said, we're not focusing on disease, or we don't have disease as an inclusion criteria. It's rather the biological principle, cryptic splice mutations, and thus we are open to any severe brain disease which has cryptic splice mutations. And we are currently focusing on 10 different diseases. You might know some of those genes. This is called BPARN. This is those mitochondrial diseases, spastic paraplegias. The use case I want to focus upon in my talk now is ataxia caused by mutations in the ATM gene. Why is this? One reason is my lab is focusing on this. The other, also biologically wise, it lends itself quite well to this N of 1 ASO approach which we are developing. First of all, in particular, the classic ataxia phenotype is quite severe. Early onset, below 5 years, quick progression. Patients are often wheelchair-bound at average year 11. So it's quite early, quite severe. You have a more variable phenotype cluster there. This might also be targetable, but then, of course, it's harder to capture, in particular on N of 1 level, whether your ASO is effective or not. But together with Rita, we are also considering to treat those patients. But apart from it being a severe disease, the second main reason why it lends itself so well to an N of 1 ASO approach, focusing on cryptic splice mutation, is that for whatever biological reasons, up to 10% of ataxia patients do harbor cryptic splice mutations, be them deep intronic or exonic. So a quite high percentage of patients does have mutations eligible to a splice modulation ASO approach. And this is verified computationally and by whole genome sequencing. And this is another reason why we chose that as a showcase for our platform. So what have we been doing? We have been, for this showcase for AT, we have been gathering patients and their mutations. We are by our registry, which I'm leading, AT patient groups, media coverage. We have now screened more than 250 ATM mutations. Out of those 850 ATM mutations, we have screened in silico to check whether it is targetable by an individualized ASO, yes or no, with standardized algorithms, which we have been developing, also together with an N of 1 collaborative, which is a US-European transatlantic collaborative for N of 1 treatments with ASOs. Out of those in silico checks, which mutation might be well eligible by an ASO, we have prioritized a certain set of mutations and taken them along the preclinical route for ASO development. And here we are now embarking on testing 4 different ASO strategies in parallel, targeting using 4 different type of ASO strategies. And we have currently developing ASOs for 11 mutations out of those 215. So we have shortlisted the most promising ones. The 4 different ASO strategies we are using, again, are not AT specific, but if they would work, they might even be applicable to other rare neurological diseases. So what are those 4 ASO strategies? It's a little bit small, I apologize for this, but it's more also for the principle. So one principle is targeting deep intronic cryptic splice mutations, where you have a deep intronic cryptic splice mutation giving rise to a new splice site, which alters the reading frame and no protein is translated. We use an ASO, so to speak, to cover up the cryptic splice mutation, thus allowing the normal pre-MRR being built and thus normal translation. A The second strategy is not deep intronic cryptic splice, but axonic cryptic splice, where you have a mutation which gives rise to an axonic cryptic splice site, again altering the reading frame, no protein, and we cover up with the ASOs this axonic cryptic splice, allowing normal protein translation. The third strategy is even more innovative, even more harder, but more patients would be amenable to this. That is not the cryptic splice, the deep intronic or axonic, but targeting canonical splice mutation. So where the canonical splice site is being weakened by a mutation, and what we are doing in SNSO principle, we are not targeting the canonical splice site per se, but rather an intronic or axonic silencer, so not relatively increasing, again, the strength of the canonical splice site, which is by a mutation being weakened. So it's a minus yields plus. So weakening the silencer enhances the conventional canonical splice site. We're trying this as a third strategy, and the fourth strategy is for exonic mutations, be them truncating, be them missing an exon. We're trying to do an exon skipping approach. You know this from Duchenne. This would lead to a truncated protein, but if the truncated protein still does have all functional domains intact, it might still give rise to a quite good disease course because the functional domains are still intact. That's, of course, the most risky, most problematic ones. So you have a little bit a gradual scale from the left to the right. It's getting more and more complex, but we are trying all four strategies. But this is likely this is the most lowest hanging fruit. That's the most complex one. Those is the array of ASO strategies we're using for ataxia, telangiectasia. Again, we're using for all those four strategies the same backbone, the same like nusinersen, so having all those biological, pharmacological, and regulatory advantages of the backbone, despite having different strategies. And the preclinical platform we are using, I was saying is we should make it scalable. So as input, we have individual patients' mutations, maybe just one patient in the world, doesn't matter. By streamlining it, you're having the same platform, you can scale it up. And the platform we are having is having the same approach for our in silico testing the mutation, for in silico design of ASOs, and then trying to show that it is restoring the function on the RNA level, on the protein level, on the biological pathway level, and you can pretty much just use, it's like a bakery, if you might say, so you use your bread and you use the same modules again, just having different mutations of input, and then test the toxicity, and then, however, have an individualized ASO as output by just using the same modules for development. And in parallel to this preclinical route, we are capturing natural history and doing this N-of-1 modeling. I'm not going into this as today, given the time course. That's a very exciting topic in itself, but not for today. But this is being done in parallel because, as I said, we don't want to start here, have an ASO ready, and then say, oh shit, we don't have natural history, we have no idea how to model N-of-1 efficacy. So we really want to do that in parallel, and also doing the biomarker work in parallel to have it all trial or treatment ready at this stage once the ASO is ready. And so is this effective or not? I cannot yet tell you, to be honest. There we have to wait a little bit, but we're getting there. So as I've shown you initially, as we're not going this phase 1, phase 2, phase 3 trial, we are much, much quicker into bringing those ASOs into clinic. And it's really like an accelerator hub. And there's one patient where the ASO was actually developed by, that is not yet ours, that was developed by Boston. We just, it's our child, a child from us, and we have received the ASO there. We have also helping that in developing, but this is a child, an AT child with an individualized ASO who is now in treatment. It's now for 2 years, actually. This child has been treated. She receives the ASO every 3 months in a named patient setting. For now, the patient is stable, yet I cannot say that it is yet an efficacy sign, a clinical one. I wouldn't go so far because he might just have a very lucky disease course, but for now he is still stable, and we need 3 to 4 more years, I think, until we have this deviation from his or her expected N-of-1 natural history progression to see whether it's effective or not. Plus, we're working on biomarkers to test and target engagement biomarkers to test efficacy in him. So he's, I think, the first N-of-1 being brain-treated patient in Europe. And in parallel, we are developing I have 3 lead ASOs for mutations of 3 other patients. They are not yet in clinic, they're still in the preclinical level, but for each of them we have a good lead ASO which has already been shown effective and non-toxic in the preclinical level. So those will be the next one in our pipeline to be put into clinics. And as you see, this is even a patient with a variant AT with this milder disease course. So we are really also trying to get to the milder disease clusters of those diseases, just opening up the sphere of what might be targetable by N-of-1 ASOs. And so it really, the vision is, you were talking about the visions earlier, say, get your personal ASO. Maybe we are once there. What is of interest is, I have been saying this is still a regulatory-wise underdeveloped country because we have very close regulations about trials, phase 1, the classical drug development. This is more a little bit an empty space, a negative vacuum. So we need, of course, it's good to have no regulations there, but also a bad thing because you don't have strong guidance. So even the EMA got interested, and we are now invited to apply for an EMA Prime program. So they're trying to develop of how can you standardize, how can you create a robust evidence, robust signs of safety on an N-of-1 level or an aggregated N-of-1 level. We have been invited to an EMA Prime protocol and using ataxia as a use case to set up those new regulatory paradigms. So I think this will set up also regulatory-wise, not just biologically and clinically-wise, but also regulatory-wise, new blueprints for Europe. And we're hoping to work together with the UK. There are strong efforts in the UK to do the similar thing. We are hoping to merge close bonds to do this together. Thank you. Thank you for our supporting networks.
So thank you very much. It was a very exciting presentation. And would like to ask for questions. So maybe one question from me first. So if you have patients with this single ASO treatment in different centers, do you try to kind of make sure that the assessments are similar in the different centers, or how do you?
That for sure. So we always have to have a little bit of a trade-off. On the one hand, it has to be individualized to the patient, but ideally, as much as possible, we would like to use the same assessment. So if possible, we would like to use the same clinician-reported outcome, the same biomarker across all AT patients, if the individual patient allows to do so, in order to aggregate cumulative evidence. Because at the end, we don't just want to treat a patient, we want to aggregate scientific evidence to make it robust. Not yet in a platform trial setting, that will likely come 3 to 5 years, but yet at least to aggregate evidence. And this is why we need consolidated outcomes as much as possible.
Mm-hmm. Thank you.
Hello, thank you for the talk you actually gave. And I wanted to ask a question, as well as sort of a double question actually. Would you consider this a form of personalized medicine in this case? And also, the other question I have is, can you actually use this type of research and expand it to all types of genetic disorders, or even streamline the process? Because rather than just going through the typical phase trials that you normally do for genetic disorders, I wondered if this research can be used for just streamlining genetic research rather than just going through the regular process for all types of diseases, actually.
Yeah, thank you for your question. Your question number one, whether I would consider this personalized medicine, I would like to say yes. It's just the term personalized medicine has been overused in the last 10 years, and it means everything. Even if you target the pathway in Parkinson's disease, where you still have 500 patients, you call it personalized medicine. So this term has been used informationally in science and the media, so this is why I don't use it, because this is a really ultra-individualized medicine, not just personalized. So it's really on a new level, and this is why I call it 2.0. It's really individualized rather than just personalized, but that's a nomenclature problem. Point number two is, what is important is, I would not say that there is a clear advantage and reason why we have this classic drug development paradigm of phase 1, phase 2, phase 3 trial. It's just not a burden or whatever. We should do that wherever it's possible. So I would not say in genetic medicine as such, we should get or need grid or try to shortcut the classic drug development paradigm. I would never say this. It is just for those ultra-rare diseases or ultra-rare treatment conditions which are not apt to this classic paradigm. There we do need a second alternative paradigm. So I would not generalize it to genetic medicine as such. However, I would generalize it to genetic medicine of ultra-rare diseases or individualized treatments where those classic paradigm does not work. And as of such, it could also work in gene therapy, it could work whatever you name it. It's also not just, it could even for small molecules. If you want, one can target a specific protein pathway or what, which is just specific to your individual, we will have this N of 1 treatment. So this idea of N of 1 treatments, that will be coming up in all of our in neurology and all of our medicine in the next years. But it should not replace the classic paradigm where the classic paradigm is still possible.
Brilliant. I think we should move on to the next talk. All speakers will be available at lunchtime to have conversations. Brilliant. So third course in the Advanced Therapies Feast. You might not need your lunch. So next is Dr. Catherine Mummery, a consultant neurologist at the National Hospital for Neurology and Neurosurgery. She leads the NIHR Dementia Translational Research Collaborative and has a mission to ensure that we have treatments that alter the course for neurodegenerative diseases, but that they can also be delivered promptly, safely, and equitably. Thank you very much.
Thank you for the lovely introduction. After those two fantastic talks and before lunch, I hope to keep you entertained. By the way, thank you to the organisers for inviting me and enabling me to play hooky. I'm supposed to be packing boxes and moving house right now. My husband's doing it on his own with the dog and is not entirely pleased that I'm here, but it's much better for me. Right, so I'm going to try and take you on a brief journey in the next 20 minutes or so in terms of what genetic discovery has helped with in dementia therapies, including the new news or developing news that we have at the moment in terms of treatments that are the first that have shown a change in the course of disease. And then I'm going to talk a little about the genetic therapies, but I am a clinician and I am a trialist, so this is going to be a very clinical discussion. And these are my disclosures. See? Nothing to hide, I promise. So hope is something that's incredibly important in all of our research, but particularly in clinical trials. As a trialist, we hope that we're conducting a trial in the right way. We hope that we're onto something with the avenue that we're pursuing. As a participant in a trial, you hope that you're on the drug rather than the placebo. You hope it's going to make a difference to you, but also you hope that you're doing something rather more altruistic that is really for the greater good. So it's a powerful part of what we do. This is a picture of Hope sitting on top of the globe, blindfolded with a lyre. There's only one string that hasn't been broken. And people talked about this picture and said, well, actually it looks more like despair than hope. And I can see what they meant. But what's countered that, saying that, thank you. What's countered that, saying this is hope, because hope doesn't actually require expectation. Hope is simply that something is possible. And in fact, when something looks like it might be almost impossible is when hope is most powerful. And in fact, in the last 20 years, I think that in dementia trials, hope is actually at times the thing that's been keeping us going. But now, I think we have a right to have some expectation about where we're going, have a right to be optimistic, not just hopeful, because of the recent developments. This is a woman who really started it all off in terms of understanding one particular type of hypothesis within Alzheimer's disease. This is Carol Jennings. And Carol, when she was a teacher in Nottingham in the mid-1980s, had worked out there was something very wrong with her family. People kept dying in their 40s, and they kept dying with a horrible dementia. And she also found out that John Hardy, who was then Dr. John Hardy, and Martin Rosser were working at Mary's looking for genetic causes for dementia. And she wrote to them, that's her letter, saying, there's something going on in my family, you need to explore it. And she persuaded every single one of her family to have a blood test. And that was the direct reason for the discovery of the first gene in Alzheimer's disease, thanks to Carol. In fact, she actually then continued to be a really powerful advocate for Alzheimer's disease and has been up until now when she's very severely affected, so unfortunately can't do anything anymore. Her son has taken up the reins, and both of them have been really actively involved in trials and advocacy, an extraordinary family. And from that came the amyloid hypothesis. We went down that route almost exclusively for 20 years, and there was a lot of debate as to whether this was the right way to go, because there were an awful lot of trials that didn't show any positive results. And the first time we saw something that might have been a signal that maybe we're at least partly on the right track was aducanumab. You will all know that story very well. It remains highly controversial. The results were controversial. The analysis was controversial. The accelerated approval was controversial by FDA on basis of reduction of amyloid, even though in the phase 1 trial it did show that you could do that and show potentially a change in the cognitive rate of decline. The drug has now been abandoned by the company. But following that, and fortunately following that, a much clearer result, because the research community was really quite polarized by that controversy, which was not helpful in progression of research in dementia. Lecanemab came along, the Eisai Biogen drug. So this was, according to protocol, completed phase 3 trial, and it showed if you drop amyloid sufficiently in the brain, as measured by PET, you do slow cognitive decline, albeit by a modest amount. It was approved fully in July last year, and hot on the heels of that lecanemab drug, in the States at least, and hot on the heels of that lecanemab drug was donanemab, which is currently under discussion by the FDA, and again, in phase 3 trial, showed very similar results, and I'll come back to those in a second. Lecanemab at the moment is under discussion by the EMA and under discussion by the MHRA, and we wait and see what happens there, but it's been given in the US, in Japan, and China. So we have the first DMT in Alzheimer's disease. That's a huge statement, but these are not cures. These just slightly slow down the cognitive progression, and we need to build on that. But it's the foundation, which is what we need in dementia. We've not, unlike epilepsy, for a very long time, we have not had that. So very briefly, some of the primary top-line results for lecanemab. What you see on the bottom right is a dramatic reduction in amyloid levels in individuals that was taken down sufficiently low for those people not to be eligible to go into the trial by the time they'd had 18 months of treatment. And you see a modest decrease or slowing in the rate of cognitive decline on the primary endpoints, so CDR sum of boxes. The secondary endpoints were consistent across a range of cognitive and functional tests, and again showed a range between 26 to 38% in terms of the rate of slowing. And importantly, that bottom right square is the burden, carer burden over time. So a 38% reduction in carer burden accumulation over time, which I think is very meaningful. The secondary biomarkers also changed, so it showed downstream changes. So again, suggestion of disease modification. Donanemab, as I said, phase 3 trial was presented last summer. Very similar drug in some ways, but a slightly different design. So in this, they showed amyloid positivity in the individuals that had to go into the trial, as they did in lecanemab, but they also stratified by levels of tau, according to tau PET. So there was this sort of Goldilocks effect of if you had too little tau, you didn't go in. If you had low to medium tau, primary analysis, and high tau, part of the combined analysis. And the reason I say that, I'll mention in a minute. The second thing about this trial was the drug was given until amyloid levels got to a certain level. When they got to that certain lower level, the drug was stopped, and they were just monitored. And that may have implications for future ways of treating, that we use an induction system rather than a regular infusion. Again, dramatic lowering in rates of in level of amyloid within the brain, both in the combined population of those two subgroups and in the low population, and associated cognitive slowing of cognitive decline. So if you look at the combined population, that's very similar to lecanemab, very similar results, around 27, 29%. If you look at the low tau population, that's an earlier population, there is a higher level of benefit. And it's possible that what we're seeing is that if you give this drug earlier, you get greater benefit. And we're starting to see that in exploratory analyses in a number of trials at the moment. We need to see a trial that is dedicated to that, and the A345 trial, it reads out next year, is amyloid-positive individuals that are early in the course of their disease, earlier than this group. And we'll see whether that confirms that you get more benefit from the anti-amyloid therapies if you give the drug earlier. And the reason that's important, back to the genetics, if you look at a genetic population of AD, you can see pathology 20 years, 15 to 20 years before people develop symptoms. So this is why I say we need to be going earlier. What we're seeing with symptoms is mid to late stage Alzheimer's disease. We are starting to learn from these different monoclonal antibodies that there is a consistency in the things that seem to have an effect. So if you look on the left, these are drug trials across a number of drugs that have been positive according to their endpoint, CDR Sum of Boxes cognitive functional test. If you look at the right, negative trials according to the same test. And there is a theme. The difference is, in the ones on the left, they lower amyloid low enough, lower than 25 centiloids, a standard measure. In the right, they don't. And this is even when, if you look here, aducanumab positive trial, aducanumab negative trial. Same drug, same trial. For various reasons we can't go into today, this one managed to lower down below 25 centiloids, and that was the positive trial. In addition, it seems that the rate of decline is also something that's important, at least in a time-limited trial. So donanemab and lecanemab both lowered amyloid much faster than any of the others. Gantenerumab is a negative trial times 2 phase 3 trials. Even though it went on longer, it never got down below 25 centiloids. Both of them were negative. So I think what we need to do is take these as the beginning and really learn from them and start to develop drugs that do the things that we know work. And one of the things that's doing that is, for example, taking drugs through the blood-brain barrier in a different way. So Brain Shuttle is a trial using one of these in an active transport system across the blood-brain barrier that so far in early phase trials has shown much quicker reduction of amyloid and no ARIA because it seems to affect things differently. Talking of ARIA, if we're going to give these drugs clinically, we have to be clear about the side effects and we have to be clear that we can manage them in our clinics. Trials are very different to clinical environments, as we all know. And this is an example of ARIA. So this is an individual who developed a left parietal area of change in signal. They were stopped on drug and it gradually resolved, and then they were restarted on drug. They were asymptomatic throughout. And that's the norm. In trials, normally with these drugs, what you see is you pick up the ARIA, which is the commonest side effect of these drugs. You pick it up on routine monitoring scans. You don't pick it up for symptoms. You ask about symptoms. Occasionally, people are symptomatic, but most of the time, 80%, they're not. And in most of them, they have it in the first 6 months, so it's critical you monitor then. Very rarely do they happen later. But you do have cases of serious adverse events and serious seizures, potentially hospitalization and needing monitoring. So we have to be able to monitor that and deal with that in our clinical situations, which at the moment, in some services, would be challenging. We also need to be aware of the increased risks, and we know that people with ApoE4 homozygosity have a higher risk of this happening than others, and we know that people with CAA or cerebrovascular disease also have a higher risk of developing ARIA-E or microhemorrhages. The critical thing is that we talk, if we have this in clinic, that we talk to our colleagues in radiology, in stroke, in A&E, in any scenario where they might be encountering people on treatments, and that patients have clear cards or other information that show that they're on treatments. Otherwise, this happens potentially. So this was an individual who developed focal symptoms, likely due to ARIA, because that's possible. She went into A&E, she saw a stroke doctor. As per protocol, she had a CT scan, not an MRI scan. So this wasn't seen. She was thrombolized because they thought she'd had a stroke. That's what happens if you thrombolize somebody that has friable blood vessels. So this is something that is incredibly important if we are rolling these drugs out in clinical services. Many questions come from the new drugs, more questions than answers. I've talked about some of these. Modest benefits only with a dramatic reduction in amyloid. Why is that? Well, perhaps we need to go earlier. What stage do we give these drugs? Again, perhaps it's earlier than we're doing that right now. And what happens when you give them over a longer period than 18 months? And there's data coming out about that. We hope it's going to be cumulative, but we wait and see. We need to see that data. Critically, patients that are being discussed with these treatments want time, but they want time when they're independent, when they're mildly affected. They don't want time when they're completely dependent on somebody and their quality of life is poor. So we have to be clear about when it's a good idea to be giving these drugs and trying to slow things down. I've talked about ARIA and the work we need to do to ensure that we predict those and minimize the severe ARIA that are very rare, but they happen, so we need to know better what we're doing there. And it's a modest effect. So should we be giving it in combination? Alzheimer's is a complex disease. So I think it's critical that we look at what happens with these drugs in combination. There are now some trials giving anti-tau therapies alongside anti-amyloid therapies. And critically, clinical services need to be fit for purpose to be able to give these drugs. And that's to remind me to pause. Firstly, to say that's the day I discovered that the world was round. Also, that's the day I discovered it's not a good idea to look at your parachute before you finish the jump. So thank God I didn't see this picture beforehand. I was fine. But the horizon is changing dramatically, and we have to grab that and discuss it with each other in terms of management of dementia. I think it's a huge opportunity, but it's an enormous change. I think some people are struggling with that conceptually. I think it's incredibly important. This change isn't going to happen quickly, but we need to be talking about it now because these things move incredibly slowly, as we all know. So I want to move now back to genetic therapies, which we've been hearing about in the past two talks. And you heard about nusinersen just now and how the backbone is being used in Matthis' fantastic work. The treatments in SMA really are a bit of a poster child for the gene silencing treatments and genetic therapies in that they have shown what a dramatic change you can actually make. And this is an example of that impact. So this is the Lee family. They have 5 children, 3 of whom have SMA, infantile SMA. And as you heard, that is a horrible degenerative disease, as you know, that can kill by the age of 2 to 4. So these children, that's Jocelyn. She was born in 2007. She died age 4. There were no treatments for her. That's her brother Nathan. He's in a ventilator wheelchair. He's 6. He's given treatment. He's stable, but not before massive damage was done to him. That's his baby sister, Kira. She's 2. She was diagnosed in utero. She started in treatment at birth. She has normal motor milestones. She has no symptoms. She is effectively cured. And that's a terminal disease by the age of 2 to 4. If we can harness any of that for Alzheimer's disease and other neurodegenerative diseases, then that isn't just shifting the needle a little bit to the left and slowing cognitive decline down a little bit. That's radically changing the course of diseases. So I think that's incredibly exciting and incredible potential. And perhaps we can use these in non-genetic diseases as well, and that's exactly what we're doing. So these are 2 genetic therapies that I'm going to talk about that are both being used in Alzheimer's disease. The first is an siRNA, it's an interference RNA that is targeting APP. You heard about APP from Carol earlier. This is a genetically validated target, and interference RNA, as you will know, effectively hijacks the cell's machinery that regulates gene and protein production and uses that to prevent the messenger RNA from leading to production of that protein. This is the Alnylam APP siRNA. It's the first siRNA to be tested in the brain. We started about a year ago, and the aim is that you reduce all products in all compartments and hopefully enable natural clearance by doing so. We started this January last year. It's a phase 1 trial. This is interim results, so these are blinded results. The trial hasn't been completed yet, and I'm just showing you data from the first 3 cohorts. And I'm just wondering, my timer has stopped, which isn't helpful. So I'll just keep going, and I'll hurry up because I'm a bit late. But it's lunch, so you're not hungry, it's fine. Sorry. So this is a single ascending dose study with 1 dose given intrathecally. This can't cross the blood-brain barrier. And this is data from the first 3 cohorts that were produced. This is in young-onset Alzheimer's disease because they have increased production rather than reduced clearance in terms of amyloid products. Okay, it's a phase 1 trial, so it's safety first, but there are some exploratory measures that we've been looking at. This data shows you the level of APP fragments in the spinal fluid. And what you see is 1 injection on day 0, and the different doses here plus the placebo, which is red. This is the low dose, this is the medium, this is the high dose. And after 1 injection, there is a dramatic down to 60 to 80% reduction in those fragments in the spinal fluid that is sustained out to 10 months. It's normalizing, veering towards the norm, but it is a sustained response out to 10 months. And associated with that, this is very early data and blinded data, associated with that, we saw a significant reduction, or a clear reduction, in levels of the precursors of amyloid plaques. so A-beta 42 and A-beta 40. Clearly, we need more work on this, and we need to finish the trial. We're now going into multiple doses, and they're going to be given every 6 to 12 months because of these sustained responses you see. You can imagine getting one dose of a drug every 6 months or one dose every 12 months is very different from something on a very frequent basis, like every couple of weeks with lecanemab. The other thing about that drug is you're reducing the production of APP and amyloid, so it doesn't cause ARIA. So there are populations that could be at risk from ARIA that this would do a much safer job in, potentially. But it's not all about amyloid. So this is all the drugs that are currently in the pipeline as of June last year in Alzheimer's disease. And you can see there's a huge variety of targets listed up the top. And finally, we're not just in amyloid therapies in phase 3. We have other drugs as well in phase 3, including anti-tau and metabolic. And I just want to mention one anti-tau therapy, and this again is this is an ASO. So you've heard a bit about ASOs already, and this one's targeting the MAPT gene. So it's trying to reduce tau production. The aim here is, unlike amyloid, where you have a head full of it by the time you have symptoms, tau aggregation closely follows your symptom progression and disease progression. So the aim is if we can reduce that, then perhaps we will prevent that acceleration of disease. We're still not certain which species of tau are toxic, so targeting all of them in all compartments is the pragmatic solution, and it seems from the preclinical work that Sarah DeVos did that that is actually safe and effective, at least in animal models. This is just a cartoon of what it does. So effectively, the synthetic oligonucleotide binds to the mRNA and leads to its destruction, so you get reduced protein levels. This is the phase 1 trial. We started this back in 2017, and people thought I was insane to be giving an ASO to people with Alzheimer's disease in 2017. It does feel like a very long time ago now. The trial was completed, the phase 1 trial was completed with the extension in 2020 and published last year. 4 ascending dose cohorts with placebo who got monthly doses initially, and then when we saw the sustained response, it went to 3 months. All were offered the chance to go into the open label extension, and the data I show you comes from both. And we have as exploratory markers, this is in mild Alzheimer's disease, CSF that was taken at every dosing, because again it's intrathecal, and tau PET that was only possible in the last 2 cohorts because of the availability of tau PET back then, and so you'll see some data on some small numbers from there. This is the top line, so CSF results and what happened to CSF total tau and p-tau. They're almost identical, so I'll just focus over here. So this is the placebo-controlled trial. Placebo group, total tau in the CSF doesn't change very much, and then you see a dose-related reduction that is significant in the dose groups over time and is sustained over time. When you give everybody the drug in the open label extension, the placebo groups and all the other groups converge to a reduction to around 40% of baseline of total tau and p-tau. But what does that mean in terms of what's happening in the brain? And CSF tau dynamics, you will know better than me, are very complex. They don't necessarily reflect directly what's happening in the brain. And we have small numbers only. So here you're seeing 2 examples of what happened using tau PET in individuals' brains that were on drug. And this is a drug that reduces production. So what you see in the placebo part with these 2 individuals is that the tau level, if anything, slightly increases. It certainly doesn't decrease. And then they go on to drug in the open label extension, and you see a clear reduction in the levels of tau in the temporal lobes. So this is early data. It's on small numbers. We need a pinch of salt to be taken with it. And we're now in a large phase 2 trial trying to confirm these results and see whether that leads to clinical changes. So what I think is that we are starting to translate all of the work that has been done over the past 20 years into impact with some of these drugs. The monoclonal antibodies are the beginning, but they are definitely not the end. They are the start of our foundation that we need to build, like they did in MS over the past 20 years. So we need to build better drugs from anti-amyloid therapies. We need to look at other targets. We need to ensure that genetic therapies are explored both in genetic and non-genetic diseases. But in parallel with that, If we don't build our services and embed research in our services, then there's no point having these drugs in the first place. And we need to be better at early, accurate biological diagnosis and having biomarkers that ensure that we can adequately triage these patients, diagnose these patients in a scalable way. And if we do that, and I'm sorry for the word personalised, Matthis, if we do that, we will get to a point where we can integrate treatments and can, to some degree, personalise those to the individual and start to look at prevention. Thank you very much for listening, and thank you to everybody that works with me.
Brilliant, thank you so much. Perhaps we have time, so there's a question right here at the front for one or two questions. Just a reminder, if you're whispering and having conversations, I think the high frequency of the whispers are sort of amplified here, so it might be quite distracting, particularly from the top tier. But yes, first question, Laurent.
Thank you very much for the talk. Is the effect of lecanemab and other such compounds homogeneous across the brain, or are there specific brain regions that are more not affected, but let's say improved by the drug? And if true, could that explain why there is a lack of overall improvement of cognitive functions?
How would you measure the differential benefit? We talk about the cognitive changes, so clearly we can't talk about those in terms of regions of the brain. From the point of view of amyloid reduction, you have a brain full of amyloid, and then you have a brain with reduced amyloid, and I'm not sure that we have a regional differential in that amyloid reduction, if that answers your question.
It does. Thanks.
This is a reflection, indeed, and just say Kath is a fantastic clinical colleague who is very helpful to the ordinary DGH neurologist. I'm looking at that picture of despair or hope, and I'm sure your department has been overwhelmed, as has mine, by contact from carers who now have despair from what's been publicised as the cure for Alzheimer's, and just a broad concern that these things do need to be broadcast in a very careful way. And of course that's media-led, but also from the charities who obviously want to encourage things to be done, but that despair is very obvious when one has a conversation with carers that actually we're still 10 years off. That's my estimate, but I don't know, you might want to comment.
No, absolutely. I think it's the point about managing expectations and the appropriate information and training and education, and that's media, that's professionals, that's charitable organisations, that's everyone. It's critical that the right messages go out. You're absolutely right. We're all seeing people that are starting to do things like they're getting their 23andMe done because they want to know their APOE4 status before they've actually had any symptoms. They're looking at their mother and saying that she had Alzheimer's at 85. I need to see you because I want to know what to do. They're coming to see you, as you say, with moderate Alzheimer's, saying, where can I get the drug from? They're flying to Brazil and picking up the drug and taking it home to wherever it is they live in another country and having the drug given to them every couple of weeks by somebody that knows nothing about what they're doing. So I think it's absolutely critical that we get that messaging right, that in this country, which at least we have a little, hopefully, some influence over how things are developed, we can roll things out safely and slowly while we are training up people appropriately to be able to diagnose and manage the drugs, and that we have these messages. And I think charitable organisations are absolutely essential to give out the right messages, saying, this is for, and I haven't got the time to show you, but the numbers that are eligible for this drug in people with Alzheimer's disease, across a number of countries, there have been audits done, and it's between 8 and 14% of those that are at the right cognitive level and have amyloid positivity. That's a small number. Firstly, we need that message to be clear. Secondly, we need to make sure all of those people that aren't eligible for the drug are not second tier, that they have adequate care that is as good, just doesn't have the drug associated with it. And that's going to mean some very difficult conversations. So I think your point is absolutely on the money.
Perfect. I think we should call an end to this session. All speakers are available for conversations during lunchtime. Please have a look at the poster sessions as well and tweet out. But thank you to all our speakers and for everyone for attending the session.